Good morning. I'm Nandita, presenting the seminar on microbial products for commercial use under the guidance of Dr. Snehata V. The contents are antibiotic, microbial polysaccharides, polyester, hormones. Under antibiotics, cephalosporin, streptomycin. Under microbial polysaccharides, xanthan. Under polyesters, polyhydroxybutyrate. Under hormone, insulin. First, we will study about antibiotics. Introduction. The antibiotic are derived from special microorganism or the living system that are produced on an industrial scale using the fermentation process. They are the chemical substance that inhibit the growth and even destroy the harmful microorganism. Antibiotics are used in many forms, each of which imposes somewhat different manufacturing requirement. For example, bacterial infection on the skin surface, eye or ear, an antibiotic can be applied as an ointment or the cream. If the infection is internal, the antibiotic can be swallowed or injected directly into the body. The large scale production of antibiotic depends on the fermentation process. During fermentation, the organism produces the antibiotic material which can then be isolated for use as a drug. The raw materials used for the fermentation, the compound that makes the fermentation broth or the primary raw material required for the antibiotic production. This broth is an aqueous solution made up of all the ingredients necessary for the proliferation of microorganisms. Typically, it contains carbon sources like molasses or soya meal, both of which made up of lactose and glucose sugar. These material are needed as a food source for the organism. Nitrogen is another necessary compound in the metabolic cycle of an organism. There are compounds such as phosphorus, sulfur, magnesium, zinc, iron, and copper introduced through the water as a soluble salt. Next is manufacturing process. It is important that sterile condition be maintained throughout the manufacturing process because the contamination by the foreign microbes will ruin the fermentation. Before fermentation can begin, the desired antibiotic producing organism must be maintained and its number must be increased by many times. In order to grow the initial culture, a sample of organism is transferred to the agar containing plate. The initial culture is then put into the shaker flask along with the food and other nutrients which are necessary for the growth. This creates a suspension which can be transferred to seed tank for further growth. The seed tank or steel tank designed to provide an ideal environment for growing microorganism. They are filled with all the things that the specific microorganism would need to survive and thrive, including warm water and carbohydrate food like lactose or glucose sugar. The seed tank are equipped with mixer which keep the growth medium moving and pump to deliver sterilized filter air. After 24 to 48 hours, the material in the seed tank are transferred to the primary fermentation tank. This diagram shows how fermentation takes place. Next is the fermentation process. The seed tank are equipped with mixer or which keep the growth medium moving and pump to deliver sterilized filter air. After 24 to 48 hours, the material in the seed tank are transferred to the primary fermentation tank. Here, the microorganisms are allowed to grow and multiply. During this process, they excrete large quantity of desired antibiotic. The tanks are cooled to keep the temperature between 73 to 81 °F. It is constantly agitated and continuous stream of sterilized air is pumped into it. For this reason, antifoaming agent are periodically added since pH control is vital for optimum growth. Acids or bases are added to the tank as necessary. This picture shows information about isolation, purification, refining, packaging and shipping. The next is isolation and purification. After 3 to 5 days, the maximum amount of antibiotic will have been produced and the isolation process begins. Depending on the specific antibiotic produced, the fermentation broth is processed by various purification methods. For example, for antibiotic compounds that are water soluble, and ion exchange methods are used for the purification. In this method, the compound is first separated from the waste organic material in the broth and then sent through the equipment which separates the other water soluble compound from the desired one. Later, to isolate an oil-soluble antibiotic such as penicillin, a solvent extraction method is used. In this method, the broth is treated with the organic solvent such as butyl acetate or methyl isobutyl ketone, which can specifically dissolve the antibiotic. The dissolved antibiotic is then recovered using the organic chemical means. At the end of this step, the manufacturer is typically left with a purified powdered form of antibiotic which can be further refined into different product types. Next is refining. Antibiotic product can take on many different forms. They can be sold in solution for intravenous bag or syringe in pills or gel capsule form, or they may be sold as powders which incorporated into topical ointment. Depending on the final form of antibiotic, various refining step may be taken after the initial isolation. For intravenous bag, the crystalline antibiotic can be dissolved in a solution put in the bag which can be hermetically sealed. For gel capsule, the powdered antibiotic is physically filled into the bottom half of the capsule, then top half is mechanically put in place. When used in topical ointment, the ointment is mixed into the antibiotic is mixed into the ointment. From this point, the antibiotic product is transferred to the final packaging station. Here, the product are stacked and put in the box. Next is quality control. Quality control is of utmost importance in the production of antibiotics since it involves a fermentation process. Step must be taken to ensure that absolutely no contamination is introduced at any point during production. To the end, the medium and all the processing equipment are thoroughly steam sterilized. During manufacturing, the quality of all the component is checked on the regular basis. These are accomplished using various chromatography technique. Also, various physical and chemical property of the finished product are checked such as pH, melting point, and moisture content. In the United States, the antibiotic production is highly regulated by the Food and Drug Administration. Only after they are certified the batch and it can be sold for the general consumption. Next is cephalosporin. Cephalosporin C can be used to treat disease and infection caused by gram-positive or gram-negative bacterial strain. Cephalosporin C is easily produced from the fungus Acrimonium chrysogenum by the process of fermentation in the bioreactor under optimum reaction condition to obtain maximum yield of antibiotic. Additional chemical and structural modification in existing cephalosporin could further enhance its antimicrobial spectrum. Today, cephalosporin and its derivative are widely used in the treatment of number of infectious diseases caused by bacteria in the respiratory tract, infection of the skin, and infection of urinary tract. The cephalosporin is one of the major biotechnological products with a total world market of about $10 billion. Next is chemical structure of cephalosporin. Cephalosporin C is a popular antibiotic because of its excellent characteristics such as broad spectrum, low toxicity, and resistant to beta-lactamase than penicillin. Chemical structure. The molecular formula of cephalosporin C shows that it consists of 16 carbon atom, 21 hydrogen atom, 3 nitrogen atom, 8 oxygen atom, and 1 sulfur atom. The molecular weight of cephalosporin C is 415.42 gram per mole with UV absorption of 260 nanometer. Here you can see the cephalosporin structure. The method of production. The cephalosporin C production is an aerobic process carried out by conventional or non-conventional fermentation method using free or immobilized fungi. Conventional mode of production. The cephalosporin C uses either surface liquid or solid state fermentation in a batch bioreactor or continuous drilled tank reactor. Non-conventional mode of production. In non-conventional mode of production, the cephalosporin C uses either packed bed or airline bioreactor with an advantage of being operated in batch or continuous mode with proper oxygenated and agitating, resulting in high yield of cephalosporin. Requirement of the fermentation process for cephalosporin production. Fermentation medium composition provides basic components required for the fungal growth and for secondary metabolite production associated with their growth. It consisted of the following components. Coming to the industrial production, the culture was propagated in the sterile medium containing these components such as sucrose, potassium dihydrogen orthophosphate, magnesium sulfate, and trace metal solution. The culture was grown for about 6 days to 7 days in a rotatory shaker set at RPM of 200. The process of cephalosporin production by the acromonium chrysogenum on the fermentation medium at 37 at 28 degrees Celsius for 144 hours is characterized by the rapid consumption of sucrose to form biomass at the beginning of the process. The pH of the medium was kept to be 7.2 and the temperature was maintained at 28 degree Celsius by passing water through the jacket. The pH of the broth is maintained by using 2 molar potassium hydroxide and 2 molar HCl. Dissolved oxygen was maintained above 30% saturation by agitation and aeration. After the depletion of carbohydrate by the slow consumption of sucrose, during which most of the cephalosporin C is produced, is called as the idiophase and observed cell growth is insufficient. Maximum cephalosporin C has been obtained at 120 hours of fermentation, while the highest cell growth occurs at 42 hours of fermentation. The cephalosporin C is separated from the liquid broth by the use of number of separation and the distillation technique in a sequential stepwise manner depending on the interactive chemical nature of the obtained product. According to the analysis, the dry cell weight was estimated by the centrifugation of 10 ml of fermentation broth, wash it twice with distilled water, recentrifuged and kept for drying at 80 degrees Celsius till the constant weight. Coming to the downstreaming process, downstreaming process involves in the isolation and purification of antibiotic produced by cephalosporin from the liquid broth. This can be done by using separation and distillation technique depending on the physical and chemical nature of product. The most commonly used method is resin ion exchange method or solvent extraction method. Finally, the purified form of the antibiotic which is ready for packaging and shipping. Next, we'll study about the streptomycin. Streptomycin, produced by Streptomyces gracius, is an active agonist gram-negative bacteria and also against tuberculosis bacteria like Mycobacterium tuberculosis. However, it is proved to be useful in the treatment of infection caused by gram-positive, specifically resistant to penicillin. It is also useful in control of plant disease caused by bacteria as it acts systematically in plants. One of the disadvantages of streptomycin is its neurotoxicity, due to which hearing impairment and balance maintenance is lost in man due to prolonged streptomycin treatment at higher dosage. In the recent times, only dihydrostreptomycin is being produced due to ready development of resistance against streptomycin. It is used mostly in the conjunction with para-amino salicylic acid, which minimizes resistant buildup in sensitive organism. Coming to the chemical structure of streptomycin. Streptomycin and dihydrostreptomycin is an aminoglycoside antibiotic and basic component which is available as hydrochloride. 3-HCl as a crystalline hydrochloride, double salt with calcium chloride, or as phosphate sulfate with dihydrostreptomycin as a hydrochloride or sulfate. Next is fermentation. Fermentation first is inoculum production. The spores of Streptomycin graecosus maintained as a solid rocks or lyophilized in a carrier such as sterilized milk, skimmed milk is an employed as stock culture. The spores from this stock culture are then transferred to the sporulation medium to provide enough sporulated growth to initiate liquid culture buildup of mycelial inoculum in flask or inoculum tanks. After sufficient mycelial growth, it is fed up to production fermenter. Next is preparation of the medium. A production of medium contains carbon and nitrogen. Glucose, fructose, maltose, galactose, mannitol, xylulose, starch can also be used as carbon source. Peptone, soy extract, wheat extract, ammonium salt, nitrates, glycine, which can be used as nitrogen source. Magnesium, calcium, potassium, boron, molybdenum, sulfates, phosphates, chloride used as mineral source. Next, fermentation. Sterilized liquid medium with all the above substance is fed to the production fermenter. Appropriate volume of inoculum is introduced into it, and the optimum fermentation temperature is the range of 25 to 30 degrees Celsius, and the optimum pH is between 7 to 8. High rate of streptomycin production, however, occurs at the pH of 7 to 8. The process of fermentation is highly aerobic and it lasts for about 5 to 7 days and passes through 3 phases. The first phase, it takes about 24 to 48 hours. The rapid growth and formation of abundant mycelium occurs during this phase. The pH rises to 8 due to release of ammonia into medium due to proteolytic activity of Streptomyces graecosus. Glucose is utilized slowly and little production of streptomycin is witnessed. The second phase, it lasts for 2 days. The streptomycin production takes place at a rapid rate without increase in the mycelial growth. The ammonia released in first phase is utilized, which result in decrease of pH to 7.6 to 8. Glucose and oxygen are required in large quantity during this phase. Next is third phase. The cell undergo lysis, releasing ammonia and increasing the pH, which falls again after a period of continuous streptomycin production. The requirement of oxygen decreases and the contents of the medium, including sugar, get exhausted. Finally, the streptomycin production ceases. A yield of 1200 and 200 microorganisms per milliliter of streptomycin is obtained. Next is harvest and recovery. After completion of fermentation, the mycelium is separated from the broth by filtration. The streptomycin is recovered by the several methods. The byproduct is vitamin B12. The vitamin B12 is produced as the byproduct which will not affect adversely the yield of streptomycin. Next is xanthan gum. The xanthan gum is a microbial exopolysaccharide. It is mostly excreted by the bacteria of genus Xanthomonas. For example, Xanthomonas campestris and Spingomonas. It comprises of repeating units of pentasaccharides, which is made up of glucose, mannose, and gluconic acid in the molar of ratio of 2:2:1. It can be produced by the fermentation of glucose, sucrose, and lactose. Various enzymes can be used for the producing xanthan gum, which include transferase, acetylase, and ketolase. The xanthan cryogels are widely replaced gelatin in the food industry and thus helping vegetarians. The xanthan hydrogel can be used as pH sensors. Aerobic fermentation of xanthan gum using chemically defined media. Xanthan gum can be produced by aerobic fermentation process. The organism used for production include Xanthomonas campestris primarily, along with the other strains of Xanthomonas. The medium required for the aerobic fermentation comprises of at least one carbon source from glucose, maltose, and starch, water-soluble nitrogen source such as peptone or yeast extract, magnesium, salt, phosphate ion, and other trace element. Also, chelating agents such as citric acid is added to maintain stability of the medium in the presence of iron and other trace element. The pH is about 5 to 5.5 to 9. Next is the fermentation procedure. The seed culture is prepared by inoculating xanthomonas in a seed culture medium. The seed culture is then added to the fermentation medium and it is incubated at 28 degrees Celsius and is agitated. Once the cell density of more than 1 gram per liter is obtained, slowly the speed of agitation is increased from 500 rpm to 800 rpm. Medium is incubated further till the cell density of more than 2 grams per liter is obtained. Once it is obtained, the fresh media is added to the fermenter and from the previous fermentation is removed. Xanthan gum produced is precipitated by using ethanol or isopropyl alcohol. It is further purified by cold press or filtration or centrifugation. Presence of xanthan gum is confined by mass spectroscopy analysis or NMR spectroscopy. Now, let us study about PHP, a great variety of bacteria which are able to produce poly 3-hydroxybutyrate as the storage material can be found in nature, for example in soil, seawater, sewage sludge or compost. PHP is therefore commonly found substance and readily biodegradable. Microbes can use PHP as a carbon source similar to sugar. PHP can be extracted from the cell and processed in the same way as a polypropylene. The properties of pure PHP are comparable to commonly used bulk plastics. Example, polypropylene, including 100% resistant to water and moisture. Another very important property is that PHP is biocompatible, which means it can be implanted into the body and will not cause any inflammation. The PHP is a storage material which is normally produced in nature when abnormal growth condition occurs. For example, if a carbon source is available but another essential component such as nitrogen is missing. Coming to the production of PHP. The manufacturing process of PHP begins with sunlight. Though photosynthesis, carbon dioxide from the atmosphere is converted to carbohydrates via sugar beets and sugarcane. These carbohydrates are the raw material for the manufacture of PHP. The PHP can be produced from glucose as raw material or agriculture waste like, for instance, molasses or material which is refined from the process of sugar beets and lactose. The fermentation process is carried out in two steps. Selected bacteria are used for pre-fermentation. The cells are grown at 30 degrees Celsius in a stream of air in a mineral media with glucose as a carbon source. The cell multiply after 24 hours and reach approximately a cell density of 20 gram per liter. This is called as pre-fermentation. The cell continue to multiply and after 40 to 80 hours it reaches a cell density of 100 gram per liter. The PHP occurs as discrete granule with a diameter of 0.3 to 1 micrometer in cell as a storage material. At the end of fermentation process, the cell has a dry mass of 80% in a form of PHP. There are two isolation processes. The extraction method. The mechanical loads destroy the cell wall and then polymer is solved in a chloroform or another solvents. The remains of the cell must be separated by centrifugation and the filtration of the solvent. Enzymatic method. The enzymes at 37 degrees Celsius destroy the cell wall. Next, we will study about insulin. Insulin is the hormone that regulates the amount of glucose in the blood and is required for the body to function normally. Insulin is produced by cell in the pancreas called the islets of Langerhans. These cells continuously release a small amount of insulin into the body, but they release sugar of the hormone in response to the rise in the blood sugar level. Certain cells in the body change the food indigested into the energy or blood glucose. The cells can be used. Insulin allows the blood glucose to be transferred from the blood to the cell. Without insulin, the blood glucose build up in the blood and cells are starved of their energy source. Some of the symptoms that may occur include fatigue, constant infection, blurred eyes, blurred eyesight, numbness, tingling in the hands or legs, increased thirst and swallowed healing of cuts. People who do not produce the necessary amount of insulin have diabetes. There are two general types of diabetes. The most severe type, known as type 1 or juvenile onset diabetes, is when the body does not produce any insulin. Type 2 diabetes produce some insulin, but it is neither enough or the cell do not respond normally to insulin. Type 2 diabetes do not necessarily need to take insulin, but they may inject insulin once or twice a day. In 1936, researchers found a way to make insulin with a slower release in the blood. They added a protein found in the fish sperm, protamine, which the body breaks down slowly. Injection lasted 36 hours. To the manufacturing process. The synthesizing human insulin is a multi-step biochemical process that depends on basic bio basic recombinant DNA techniques and an understanding of an insulin gene. DNA carries the instruction for how the body works, and one small segment of the DNA, the insulin gene, codes for protein insulin. The manufacturers manipulate the biological precursor to insulin so that it grow inside simple bacteria, while the manufacturers each have their own variation. There are two basic methods to manufacture human insulin. Working with human insulin. The insulin gene is a protein consisting of two separate chains of amino acid and A above B chain that are held together with the bonds. Amino acid are the basic unit that build up all the protein. The insulin A chain consists of 21 amino acid and the B chain consists of 30 amino acid. Before becoming an active insulin protein, the insulin is first produced as pre-proinsulin. After pre-proinsulin, the chain evolves into proinsulin, still a single chain but without a signaling sequence. Then becomes the active protein insulin, the protein without section linking the A and B chain. At each step, the protein needs specific enzyme to produce the next form of insulin. Starting with A and B. One method of manufacturing insulin is to grow the two insulin chains separately. The manufacturers need the two mini genes, one that produce the A chain and one that produce the B chain. Since the exact DNA sequence of each chain is known, they synthesize each mini gene's DNA in an amino acid sequencing machine. These two DNA molecules are then inserted into the plasmid, small circular pieces of DNA that are more readily taken up by the host DNA. Manufacturer first insert the plasmid into a non-harmful type of bacterium E. coli. They insert it next to the LAC-Z gene. The LAC-Z encodes for 8-beta-galactosidase, a gene widely used in recombinant DNA procedure because it is easily to find and cut, allowing the insulin to be readily removed so that it does not get lost in the bacterium DNA. Next to the genes in the amino acid methionine, which start the protein formation. Plasmid enters the bacteria in a process called transfection. Manufacturer can add to the cell DNA ligase, the enzyme that acts like glue to help the plasmid stick to the bacterium's DNA. Bacteria synthesizing the insulin then undergo fermentation process. They are grown at optimal temperature in the large tank in manufacturing plants. The millions of bacteria replicate roughly every 20 minutes through the cell mitosis, each of which expresses the insulin gene. After multiplying, the cells are taken out of the tank and broken open to extract the DNA. One common way this is done by first adding a mixture of lysozyme that digests the outer layer of the cell wall, then adding a detergent mixture that separates the fatty cell membrane. The bacterium's DNA is then treated with the bromide, a reagent that splits protein chain at the methionine residue. This separates the insulin chain from rest of the DNA. The two chains are then mixed together, joined by disulfide bond through the reduction reoxidation reaction. The oxidizing agent is added. The batch is then placed in a centrifuge, a mechanical device that spins quickly to separate the cell component by size and density. The DNA mixture is then purified so that only the insulin chain remains. Manufacturers can purify the mixture through the several chromatography or separation technique that exploit difference in the molecule charge, size, and affinity of water. The procedures used include an ion exchange column, reverse phase high performance liquid chromatography, and a gel filtration chromatography column. The manufacturer can test insulin batches to ensure none of the bacterium's E. coli protein are mixed with the insulin. They uses a marker protein that let them to detect E. coli DNA. They can then determine that the purification process removes the E. coli bacteria. Next is the pro-insulin process. Starting in 1986, the manufacturer began to use another method to synthesize the human insulin. They started with the direct precursor to the insulin gene proinsulin. Many of the steps are the same as when producing insulin with the A and the B chain, except in which the method the amino acid machine synthesizes the proinsulin gene. The sequence that code for proinsulin is inserted into the non-pathogenic E. coli bacteria. The bacteria go through the fermentation process where it reproduces and produces insulin. Then the connecting sequence between the A and B chain is spliced away with an enzyme and resulting insulin is purified. At the end of manufacturing process, the ingredients are added to insulin to prevent the bacteria and help to maintain the neutral balance between acid and base. The ingredients are also added to intermediate and long-acting insulin to produce the desired duration type 1 type of insulin. This is the traditional method of producing longer acting insulin. The manufacturer added ingredients to the purified insulin that prolonged their action such as zinc oxide. These additives delay absorption in the body. Additives are Additives vary among the different brands of the same type of insulin. Coming to the quality control. After synthesizing the human insulin, the structure and purity of the insulin batches are tested through the several different methods. The high performance liquid chromatography is used to determine if there any impurity in the insulin. Other separation techniques such as X-ray crystallography, gel filtration, and amino acid sequencing are also performed. Manufacturers also tested the vial packaging to ensure it is sealed properly. The manufacturing for the human insulin must comply with the National Institute of Health procedure for large-scale operation. The United States Food and Drug Administration must approve all manufacturing insulin. On a brief account, I discussed microorganisms which are useful for the human welfare. These are the references for this section. This is the end of my seminar. Thank you.